So Sensei, the class is yours. So you can start the lecture.
Yeah, thank you. So last week I talked about fermentation food, and I didn't explain about the world's three smelliest fermented food. So this kind of food, okay? And that is kiviak. Oh, this is the kiviak. I never eat. And honhe, honhoe, and Korean food. And this is shoestring. I forget, in North Europe, Sweden, or something like that. I'll explain. Anyway, these three are believed the most the smelliest fermented food, especially in Japan. These are very famous. So, honhe will be made from this kind of shark. And shark have a lot of urea in their body. Do you know why? Maybe you don't know. Because very, sharks are very old fish. So they adapted to the salt concentration in the sea. So that means salt concentration increasing gradually. But all in ancient day, they live in low salt sea, and they adapted using urea in the body. And here they are ancient fish. So anyway, osmoregulatory function, it depends. Because of urea, they have reach. So this fish is cut and ferment. And this fish contains many urea, so smell is like ammonium. And I ate this in Korea. Yorai-san, you didn't. Indeed, I brought back for student. So you are not there. Anyway, this smell is not so bad. Just ammonium. So you just go in toilet. That's all. Next is shoestring. Shoestring is very strong smell. And here, probably you cannot see. That means shoestring is prohibited on the bank of the oiled river. So this is presented. So some people eat this in riverside because we cannot eat this in room. Very strong smell. So this is a Swedish embassy in Japan. We have the vending machine, and they sell shoestring. So if you come to Japan, you have to go to Swedish embassy in Japan. Then you can buy shoestring. Shoestring is fermented in can, and they have no sterilization. So shoestring become like this. You see, can become bigger. So this is because of fermentation. This is the video. Oh, where? How? Why? Okay. Why? Do not work. I don't know. Just a moment. Oh, sorry. Sorry. This becomes small. Should string like this. And I am opening this in the draft. You have draft? Without draft, we cannot open. And inside is like this. So many fish, fermented fish in the can. Like this. Taste is not so bad, but smell is too bad. And in Sweden, opening can indoor is prohibited by law. So you cannot keep in your room. That is another strange food in the world. This is a method how to cook. So they have no sterilization. Generally, cans are sterilized, but this is not sterilized. So, but after fermentation, almost all microbes are dead. And here, it is usually imported by ship. Sorry. Okay. Because once you bring this into airplane, it may be broken. So you cannot survive in airplane. So probably in Indonesia, you also import streaming. Please try. In my laboratory, Hasegawa, you know Hasegawa, he bought and we opened. Okay, so please try. This is fermentation food. Next, I never eat this. So, sea bag. At the moment. Oh, I cannot see where. Anyway, I forget the name of this. So anyway, these sea bag was put in the body and ferment in under the soil. This is done by Eskimo in Canada and cold place. And you can eat this like this. Right? Not, sorry. Right? drinking the inside, like
Ah,
that. This is a this is a small, smallest food probably, but I never eat and I never seen this. If you have chance, please send me. So anyway, we have this kind of food, and this is a part of the local culture. Do you know what is this? This is Christmas present made of by this. So this is Christmas, can be a Christmas present in Canada. So,
oh.
so it depends on region. For me, this is spoilage, but for Canadian, this is a very important present. This is a culture food. And okay, and oh, well, next. Today is the second. Indeed, I cannot see upside. Oh, 違う。国のための表示。違う。No, sorry. I don't know how to manage this. Anyway, uh, today I will talk about mainly about koji. Koji is a Japanese traditional technology relating to using microorganisms. Here, koji. Please remember koji. And miso, soy sauce, sake will be explained. This is a review of the last uh, last week. Please don't forget this. Lactobacillus is lactic acid bacterium is very important for us. Probably humans have adapted to microorganisms that survive in acidic environment. This implies a symbiotic relationship with lactic acid bacteria. We may say about lactic acid bacteria. And about yeast, except your country, yeast was domesticated. Because in Japan, this is very important and ferment and making bread and something like that. And why? Sorry. This is sorry, this is my sound. Sound we don't need. This is how to make how to eat narezushi. Narezushi is like this. Oh, sorry. This is narezushi. Put in the bottle. Not bottle.
Like a basket, sensei?
Yeah. Sorry. And they are my classmate when I was high school. I asked them to eat and see the taking the video. This is an Arebushi. This is TV program in Japan. You can see. Oh, thin. Now it's become big. Okay. Next. So, mhm. Oh, okay. So this is a asparagus, and koji is made by cereals and asparagus, and many kind of species we have. And using koji, we make sake, soy sauce, miso, and some spirit. And that is very important in Japanese culture, indeed. Here, sake, shochu, aomori, and soy sauce, and miso, and they have special asparagus, fungus. Sake is orize, and shochu was luchiensis, and awamori is kawachi, and soy sauce is soya. Probably came from soy sauce, soy. So it is like this, and generally shochu and awamori was made by these asparagus, and these asparagus are black and similar to asparagus niger, kind of asparagus niger, I think. The orize is kind of flavors. And soya, I don't know well. And the role of koji is very important. Why we use koji? Here is the role of koji. This is a method how to make beer. For making beer, saccharification process is very important. After that, yeast will ferment and produce ethanol. So starch was degraded to glucose and ferment to alcohol. So koji plays a role for breaking starch or proteins. So high molecular weight biomaterial was degraded by koji. So koji plays a role for breaking. Okay. This is also video. Why? No. Oh.
Al siguiente elemento presentado por Granada.
I don't know what they are talking, but anyway, this is the theme. Now we got UNESCO recognition.
Se adopta.
Anyway, Japanese technology, maybe ancient technology using koji was listed as I cannot see UNESCO recognition. I think in Indonesia, you have many UNESCO recognition. So Japan also, Japanese food also UNESCO recognition. What should I do? Check it. Okay. The UNESCO list of intelligible culture heritage from 2024. So Japanese traditional sake brewing using koji has, I cannot see. Anyway, UNESCO this kind of things. And now in Japan, push these alcohols. This is kind of rice wine and spirits using wheat. And this is also spirit from rice. And this is seasoning using koji fermentation. Indeed, I didn't know this is made by koji. So we have three kind of alcohol, sake. And these three are called national sake. You see wine brewing, how we brew, ferment wine. You just break grape and put it in, I don't know, bottle. And you, maybe sometimes you put yeast and fermentation. And after that, we can drink wine. Very easy. But if you see the Japanese sake production, you will be surprised. This is a method. It was very difficult to write. So you can see many arrows. Very complicated method. Koji starter or yeast, lactic acid bacteria, and this become a starter culture preparation, something like that. Indeed, I need several times to confirm this is correct when I made this picture. So we call this multiple parallel fermentation. Three-stage brewing or three-stage parallel fermentation. In the case of beer, we say single-stage double fermentation. So very complicated. But if you see video, you will understand how to cook, how to make.
My name is Samantha. I'm about my brewery. My brewery is located in Gifu City. Gifu City is very nice place, and Gifu City have a rich environment, nature, and rice field. So please come to my brewery. Okay, 10 here and so huge here. They made tank in the room. Our sake is named Daruma Masamune. Daruma is never give up door because This is Dalmadol. Pour over. Always rice. That's all. This is never give up though. It's January 3rd. Please come to temperature. It's 7 degrees. Japanese sake fermentation is under cold season, and the ingredient of sake is rice, steamed rice. So next time I'll introduce making sake. So see you next time. Bye bye.
So sake is made like such a species, okay? And she said her factory is in Gifu. Gifu is a city. I and Yohane-san lived, and we are in Gifu University, and probably close to Gifu University, that factory.
Sake, or Nihonshu, born from Japan's outstanding
This is also
water
video
and the wisdom of generations.
in English. This is long. 10 minutes, maybe.
Rice is itself Japanese, and Nihonshu is a precious part of daily life and Japanese culture. Knowing Nihonshu means connecting with Japanese culture. We see the wisdom of our forefathers in traditional brewing techniques.
This is koji. This is koji making.
Born. These are sake brewing craftsmen, known as kurabito. The person who directs and is responsible for the work of the kurabito is the toji. Work in a sake brewery begins with steaming rice in the morning. Special sake brewing rice is steamed in a vat called akoshiki. Preparations for steaming began the day before. Under strict supervision of the toji, the kurabito watch the rice and allow it to absorb water. This is a delicate process that calls for the highest level of technical skill. Because the amount of water that the rice absorbs greatly affects the quality of the final product, the koji measures time down to the second during these steps. It takes about an hour to steam the rice. Here, the condition of the steamed rice is checked by squeezing a handful of rice into a putty-like form called mochi. The steamed rice is quickly cooled. Main process of sake brewing starts here. The three main pillars in sake brewing are: first, making koji; second, preparing the yeast starter; and third, creating the fermenting mash. The most important of these is making koji. Steamed rice is carried into a special room, the koji mudo, where temperature and humidity are kept high. Koji mold spores are sprinkled onto the spread out rice. The rice is wrapped up in cloth and allowed to sit for a full day to let the mold propagate thoroughly. The next morning, the clumps are gently broken up and divided evenly into special wooden trays called koji buta. The rice is thoroughly mixed to ensure that the mold will propagate uniformly across all the rice. The work performed inside the koji mudo calls for great focus, concentration, and patience.
It seems already dried, but not dried. They are growing to inside.
From this careful attention, at last, the koji is ready. Making sake requires high-quality, pure strains of yeast that will be allowed to greatly multiply and play the major role in fermentation. The koji, created in the last step, is added to water along with steamed rice and yeast.
So koji, yeast, and rice was added. This is the first stage.
The rice is cooked rice, sensei?
Steamed rice.
Or steamed rice?
Very This is very high technology.
Mm.
They don't use heater.
Yeah, heater.
We still use these techniques.
Mm.
The yeast starter is transferred to a larger tank, and more koji, steamed rice, and water are added.
So, koji and
The next day is known
rice
as
was
the
added. This is the second stage.
However, comparatively
And this is the third stage.
again. On the fourth
How
day,
long you ferment this one?
water are added.
How long you ferment?
Roughly half of
I don't remember well. Please say
this is the end of the fermenting.
please see. Probably they will explain.
From this point on, the moromi will go through many drastic changes. The koji will convert the starch in the rice into glucose, which the yeast will then use to create alcohol and carbon dioxide. The conversion of starch to sugar and sugar to alcohol takes place in parallel and in the same tank.
This is
known as
important.
multiple parallel fermentation and is unique to sake.
Mm. Mm.
The surface of the moromi, which has changed many times, at long last is quiet and still.
Mm. Mm. Mm.
The fermented moromi is placed into cloth bags, and the completed sake is separated from the remaining rice solid.
Mm.
Okay, like that, sake is made. And my student, for example, Moriyama and Hasegawa, Yorani-san knows them. They try to make sake. And I taste. It was strange, and suddenly I went to toilet. So it is not easy to make good taste sake. For student, it was very difficult.
Mm.
So that's why sake brewing is very
Expensive?
important.
Mm.
No, sake is not so expensive.
Oh, not so expensive.
But as a Japanese technology, this is very important. So about 40 years ago, Japan was famous for fermentation. Indeed. That is because of we have the technique of making sake.
Okay.
Now, many fermentation are carried out in Indonesia, Thailand, I think. I saw the factory in Thailand. Thailand make many fermentation foods and many yeast and many things. Anyway, this is a real fermentation process, but if you see the video, you can understand how to make sake. So point is koji and yeast
Yeast.
and rice. And we need pasteurization and become a sake. This is Japanese. And I didn't talk about rice. Indeed, we have to polish rice. Okay, rice polishing is very important. So rice is like this, and here many nutrients, carbohydrate, proteins, fat, ash, vitamins are here. And they have white core. Here is only starch. Pure starch is inside. And we polish like this. If you use this, this is a good grade sake. If you use this cheap sake, so how polish is very important for sake. But if you see focus, no, from the view of yeast, for yeast, very hard conditions to survive and to produce alcohol because no nutrient. So alcohol is a yeast tears or sweat, probably. So very hard conditions for yeast, it is said. And about 20% of alcohol production. There is not so high alcohol containing drinks, except spirits. Spirits is not direct fermentation. So this is fermentation of sake. Next is miso. This came from China. Origin is in China. And in the case of miso, we use koji. And koji was made from rice or wheat or soybeans. Anyway, main was soybeans. And the koji was made different way. And we have many kind of miso. This is a miso soup. Have you ever eat? Student, they know?
Maybe they know it because every Japanese restaurant they give it for free.
Oh, you have Japanese restaurant?
Yeah, in here we have a lot.
Oh, yeah.
And they are using mirin too sometimes in some restaurant. And in that restaurant they couldn't get halal registration number because it's not halal because they put it in the rice, maybe rice and say mirin. Yeah.
I remember that I forget Ramadan,
Ramadan, yeah.
and after sunset,
Hmm?
many Indonesian students, especially girls,
Huh?
went to sushi bar.
Oh, yeah, yeah, yeah.
That was very interesting.
Mm.
Sushi is halal. So for Indonesian people, it is easy in Japan if you eat sushi.
Yeah, yeah.
Sushi is very expensive.
Oh,
Yeah. Oh, miso. Okay, sorry, miso. Probably miso came from China. Jian. 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 It is said Jian of ancient China and probably came to Japan. Miso has unique production method depending on the region. So it depends on the regions. So miso can be classified into rice miso. So rice miso means rice koji miso and barley miso and soybean miso. Anyway, that means how you made koji. Yes. So this is a procedure. Soybean, salt, and koji. So which kind of koji is important? But here this didn't explain. But this was made by AI. How to make miso in English. But anyway, you will see the video again. How to make miso. This is not essential procedure. It depends on family. Anyway, breaking. This is rice koji, and we call this rice miso. They have to make anaerobic condition. So removing the air. For pasteurization, to kill microbes. Six months. So that is how to make miso. And this is a map of Japan. And we have many kind of miso. Barley miso, wheat miso. Wheat is okay. And sweet white miso in middle of Japan. And dark soybean miso. I think Yoani-san eat this. No?
Dark, so I think yeah, because I think that there is a miso that halal in Japan, so
Yeah.
sometimes I eat it.
Yeah.
Mm. But
This
when
is
when I stay in Japan, I don't
yeah.
like miso. But when I stay in Indonesia, I like miso so much. And my
Yeah.
daughter really like miso.
Oh, really?
Mm.
It's kind of miso.
I don't know. I just get a free from that restaurant, so
Yeah.
my daughter is really like miso. Mm.
Oh,
I should bring her to Japan to
yeah.
eat miso.
Yeah. And yellow balanced rice miso and salty rich rice miso. In this area, many people like salty food and they become sick easily. So they are changing now. And in the north place, this is Hokkaido, depends on families. Because about 200 years ago, there's just forest. And many people come from the main island. The microbe is Aspergillus oryzae, or it depends, and Tetragenococcus halophilus. Halophilus, you know, salt-like. This is a lactic acid bacterium. And Schizosaccharomyces leuciscii. This is also salt-like, salt-resistant yeast. So anyway, fungus, lactic acid bacterium, and yeast.
Yes.
Yeah. So similar to sake.
Sake?
Yes. This is a kind of food made by miso.
Mm.
Like this is pickles, and this is miso soup. And this is special in north of Japan.
Mm.
This is kind of rice bowl, and put the miso. And we have miso bread. Next is soy sauce. Soy sauce is special in Japan. And it was made in Yuasa town in Wakayama Prefecture, where I was born. So here, I was born here, and here, soy sauce was also born. Around this century. Anyway, it was miso, but there is some liquid, and that was used for as soy sauce. So for making soy sauce, you just put water. So they put for making miso, they put salt. But for making soy sauce, you put salty water. That's only, that's the difference. That's all of anyway, there's not so big difference. But you have many soy sauce, dark, light, rich, fermentation, and white. And this is also depend on regions. And see, this is fermentation of soy sauce. It is very strange place, but they don't put microbes except koji. They have to put koji, but after that they don't put microbes. Do you know why? Do you,
Because they already have the microbe there.
yes, microbes are here.
Yeah.
Yes,
Ah.
you know. So dropping.
Oh, dropping.
Yeah. So many microbes around here and dropping. And after fermentation, we have some bubble and go up to here and maintained. It is like this.
Mm.
And here is many microbes. And going up and drop down. And indeed, when I come to here, I took some parts, secret, and taste. It was very good. Secret. Anyway, this is indeed we make soy sauce in factory, but we also have traditional spaces, production places in many places, and especially in Wakayama Prefecture, we use these. Oh, so that is fermentation, and we have focus on science slightly. So Yorani-san knows well, and Tifa-san? No, who?
Diva-san.
Difa-san knows about aflatoxin. So asparagine had aflatoxin-related genes, and it may produce aflatoxin. About 15 years ago, asparagine gene was sequenced, and we found that they have aflatoxin-related genes. And many scientists wondered, oh, it's okay or not. So many scientists focused and studied because they have gene. Why they don't produce? And now we know they have the structure of gene, but some regulation does not work. So Aspergillus flavus and Aspergillus oryzae, both Aspergillus have aflatoxin-related genes. But Aspergillus oryzae cannot produce aflatoxin because they don't have the regulator genes. That's all. So flavus and oryzae may be same species. But historically, they were several years later, they may lost. Indeed, Aspergillus flavus do not produce aflatoxin under laboratory conditions. Indeed, it is said. You saw.
Yeah. DIFA research is a fail too.
Yeah.
Maybe
Yeah.
DIFA you should study here and you should listen Professor Iwahashi carefully and then you get the answer.
Yeah, no answer because you will find, DIFA will find that will make history of microorganisms. So once you find why they don't produce, you will be the top scientist in the world. So you have the chance. DIFA has the chance.
DIFA have a
Don't-
chance.
Don't forget.
Yeah. Under my supervision.
Yeah.
And then I get Nobel Prize.
Yeah.
DIFA you should.
You get? DIFA cannot, but you will get.
Yeah,
Oh,
DIFA you should study hard and help me to get Nobel Prize.
good. So how about the type culture?
Mhm.
You know type culture.
Yeah.
Your culture is type culture or you isolated?
We isolated
Ah,
Mhm.
indeed.
wild type,
Yeah, wild type, but not type culture.
not type culture.
But probably you may continue to isolate.
Mhm.
So maybe anyway, indeed, if you get type culture from some organization,
Hmm?
they produce aflatoxin. Only a
Yeah.
first few times. So 2 or 3 times they produce in general medium. But gradually they don't produce. We don't know how, why. So maybe they have no contamination or no other microbe attacks Aspergillus. I don't know. We don't know. But if you find why, you will be invited to Japan. Because for Japanese, Aspergillus oryzae is national microorganism. And this is a pathway, complicated indeed. So maybe because of this pathway, there is in some cases, there oxidation and reduction. So maybe that is protect or something like that. Please be careful about the pathway
Mhm.
and genes. And my wife told me that they produce messenger RNA
Mm.
after 2 or 3 times. They still make messenger RNA,
Mm.
but they don't produce. So maybe some conditions, but I don't know. Anyway, this is like chickens. Chicken cannot fly. So Aspergillus oryzae is domesticated from Aspergillus flavus. And we don't know, but this is a very good target for studying, indeed, I think. And please take care about aflatoxin. In my wife's senior, 3 generations died because of cancer. So you have to take care, especially liver cancer.
Yeah.
Anyway, taking care is very important. Okay. And what I want to say is, DIFA-san's target is very important, especially for Japanese.
Yeah,
You will be Japanese hero, heroine.
yeah.
If you find, don't tell Yorani-san, tell me. This is just information. I have time, so I'll tell you. You know, D form of amino acid and, sorry, L form amino acid and D form amino acid. We know. You know? Student know? Okay,
Maybe no.
okay. And generally we use L form, but microbe sometimes produce D form amino acid. And if you compare D form and L form, completely taste is different. For example, glutamic acid, L form is umami, good taste. And alanine, middle is sweet, but if you taste, it is very strong sweetness. Did you taste in Japan? No.
No.
No. Okay. I bought D form and I taste in laboratory. It was very sweet.
Mm.
So maybe microorganisms change the taste because of, maybe one of the reasons can be this. Or sometimes if you eat fresh vegetable or fresh fish, that is sweet. That maybe come from this reason.
Mm.
Okay, that's all.
Thank you very much, Sensei. For a student, do you have any question related with the lecture? You can raise your hand or you can write your question in chat column. Any question? Yeah, Diva?
Okay, Sensei, can I see your about rice polishing?
Rice?
Rice poisoning.
Polishing.
Oh,
sorry.
polishing. Oh, okay.
Polishing. What is polishing?
In slide sake.
Hi.
Okay.
Sake. Oh. Auto. Yeah.
Next.
No.
Lah, nih. Okay. So in this.
No.
Yeah, this one.
Okay.
Yeah. So in this picture, I curious, is it in Japan? Sake, is it classified in premium or regular sake?
You mean which is a regular sake?
Japan have,
Many kind of sake.
but what? Okay. What's difference
Between the rice polishing? Do you mean Diva?
Yeah, it means like 70% or
Ah,
70% grade of
yeah,
sake.
yeah,
Yeah.
yeah.
Can you
So
catch my?
if you drink, yes, you cannot drink this, so I will explain. This is pure alcohol, like that. Taste is pure alcohol.
Mhm.
And this is taste like here. So you feel here's taste.
Yeah, I know it.
You understand?
Like how can I say that? Like the taste is like how can I say that? The raw material, raw rice. Yeah.
Yeah.
And Difasang asked the premium one or which one is the best, Sensei?
Oh, of course, here.
Ah, does one how many percent alcohol, Sensei?
Alcohol percentage is similar.
Oh, similar.
Yeah, but taste is different.
Ah, okay.
And this is pure,
Mm.
like a white wine, similar to white wine.
Mm.
Maybe you don't know.
Yeah, yeah. Any question? Another? Diva, did you catch the answer?
Yeah. Thank you, Sensei.
Okay, thank you.
Any question from student? Yes, Rendra.
Thank you.
Yes. 申し訳ございません, Sensei. I'm just curious how long it takes that to make a sake that, I mean, like one batch, how long it takes? I mean, the time is that 3 months or more? I think that's my question.
Totally, yeah. Totally from 1 to 2 months, I think.
Mm.
It
Yeah.
depends on places.
Mm.
And
So,
slowly is better, it is said.
Mm.
so that takes 2 months. Is that depends on the weather? Like if
Yeah.
summer, it's more like
Yeah.
difficult, or
Yeah.
it
So,
doesn't matter?
yeah, we have to make ferment in winter.
Yeah.
And the winter temperature depends on the places, so it depends.
So is that any season that stop produce sake?
Hi.
Like
Summer, we don't produce.
oh, yes.
Is it in
Okay, that
summer? I'm so sorry, I didn't catch the answer. Is it in summer? Do you how can I, do is it in Japan still produce sake, Sensei, in summer?
No, just drinking.
Oh, and you just produce it in winter.
In the winter.
Winter. Ah.
In
And,
fly, we have the rice.
hmm.
And in winter, we produce sake.
Oh, okay.
In summer, we drink.
Okay. Yeah, I see it. Ah, okay.
So, and cold condition is better, it is said. So we
Mm.
have to make stress to east
Yeah.
for producing tears
Fierce
and
and sweet. And then,
yeah,
Sensei, how can I say that? In that sake, you have the rice,
yeah,
koji, and another, and you have the liquid one. For
yeah.
sake is only for liquid one. And what happened with how can we call it residue? How can I say that? The rice one. Do you make it something for that?
You mean supernatant and
Yeah,
precipitate.
and the supernatant and precipitate, and the liquid, the supernatant we drink it, and then
Yeah.
how about the precipitate?
Precipitate can be used for making pickles,
Oh, pickles.
and sometimes we eat.
Oh, okay. Yeah. Okay. I see it. Sensei, I saw in miso production, not
Yeah.
miso production. I forget it. There is a home that you show it us. You accidentally put a bug there.
Huh?
Yeah, you accidentally put bug there. If there is a contaminated there, what will
Yeah.
happen? In one home, they get
Yeah.
contaminated. How can they remove the contamination?
So they take care to avoid airs.
Yeah,
If
but
you
if
so if you made a mistake in Japan and contaminated, your stepmother told you go out. That's all.
yeah. But how can I say? If that's one, if I am foreign people, but if the Japanese did it, how can it? Do you what will you do?
Probably just for throughout. We don't eat.
Okay. So how can you remove the contamination?
We once contaminated, probably we don't eat.
Uh,
But maybe there are some people, but as I know, fungus is very dangerous.
mm, okay. Any
So
question from student?
if you put many salt, we don't make mistake so much.
Oh, okay. Yeah. Yeah,
Oh,
Marvin, you can ask the question.
Okay, thank you, Mr. Iwan, for the opportunity. So good afternoon from Indonesia, Prof. Iwahashi. So my question is, as you explained before about the Aspergillus oryzae and Aspergillus flavus are closely related in genetic aspect, right? So my question is, if environmental conditions unfavorable, is it possible for Aspergillus oryzae to produce toxins, or is there any condition that can make the Aspergillus oryzae produce toxin?
Yeah.
if
Yes.
you do that in Japan, you will be killed.
Yeah.
You understand? Japanese believe they will not produce.
Oh, okay.
No.
Is it
And
the
we have no.
environment?
Part,
How about the environment? Malvin
pardon?
asked it. The
Huh?
environment, if the environment is good to produce aflatoxin, is it they
No,
can produce? Oh, they can do it. They couldn't.
because they don't have regulator genes. It
Mm.
is safe.
Yeah.
Japanese scientists always tell.
Mm.
And Ministry of Agriculture announced Japanese sake is safe.
Safe. Mm.
If you find aflatoxin, you will get a lot of money.
Oh,
We
you
found
can.
the condition, and we have to find, if you
Yeah.
say.
You can
But,
climb it.
but anyway, indeed, according to the sequence, they cannot produce. They just have structure gene. Indeed, it is very difficult to produce aflatoxin using Aspergillus flavus. Not only oryzae.
Yeah. In DIFA experiments, DIFA get the Aspergillus flavus from kombucha, and right now she tried to how can I say that? She tried to ferment it using Aspergillus flavus, and already 4 times she did LCMS to detect aflatoxin, but she couldn't get it.
Yeah.
Mm.
Gifa-san isolated from kombucha.
Not DIFA, but DIFA,
Sama.
yeah, DIFA senior.
Yeah. So that will already be origin.
Maybe
Easily.
already many time. Yeah, many
Yeah,
time
so
culture.
because that's a commercial product,
Yeah.
they continuously grow.
Mm. Okay.
So you have to better find the type culture that produce aflatoxin. Indeed, in Japan, there is such type culture promising this produce aflatoxin.
Yeah.
Maybe you can find that in DSM, German or Japanese such organization. But probably it is only first and second. My wife told me. Or you can check messenger RNA
Yeah.
from your strain, species
Strain.
strain.
Okay. Yeah. Any question from student? Any question? Okay. I think that is already finished
Yeah.
for today's section. Thank you very much, Sensei.
Thank you.
Thank you very much for the
Thank you
lecture.
for listening.
Yeah. I hope that every student can listen and can study a lot from Iwahashi Sensei. And if you have any question, you can directly ask to Iwahashi Sensei. But for today's section, because already finished, we can continue the section or the lecture maybe 2 weeks later, right, Sensei? 2 weeks later.
Yeah.
And for a student can that's the student who take the bacteriology and food hygiene lecture. Yeah, you can for next week. The class is with me, not with Professor Iwahashi, so we can directly have meet the class. I mean, not online. Thank you very much, Sensei.
Thank
Thank you
you.
for the lecture.
So after 2 weeks, 3 weeks later.
After 2 weeks, maybe. I'm already.
Yeah. It's
Yeah.
okay.
Okay, Sensei.
It will be hard for me. I didn't prepare.
Okay, Sensei. Thank you very much, Sensei.
Thank you very
So,
much
guys,
for
thank
listening.
you very much.
Thank you.
Mm.
See you.
See you, Sensei.